Okay, so just before we get into it, remember the term somatic autonomic refer to divisions of our peripheral nervous system. And remember, the peripheral nervous system consists of everything except for the brain and spinal cord, essentially. Okay.
Isn't what we just talked about?
You know, we introduced this last week when we talked about
we just
divisions.
talked about it in anatomy too, right?
Yeah, absolutely. We've been
okay.
talking. There's definitely overlap between the two classes. So when we're talking today, we're going to be talking a lot about nerves. Okay, I really shouldn't say a lot, but a decent amount about nerves. Okay, it's not the first time we've talked about nerves. On our last lecture, the very end, actually we didn't get to it on Thursday, so I recorded it and posted that for you guys. Hopefully you watched it over the weekend. We discussed the 12 pairs of cranial nerves. Okay, 12 pairs, that makes 24 total, which carry signals to and from the brain. And then what we'll kind of focus more on in this lecture are the 31 pairs of spinal nerves that arise from the spinal cord, so 62 total. Now, definition of a nerve is a cord-like organ composed of both sensory and motor neurons bound together by connective tissue that can transmit action potentials in opposite directions. So you can think of it like a two-lane highway. If you did watch the video on the cranial nerves, we see that it's not always the correct way to describe nerves, because there's actually three types of cranial nerves. There's the mixed cranial nerves, which are like the two-lane highway, but then there's also what we call sensory nerves, which are predominantly made up of sensory neurons, which means that they carry action potentials into the brain. Okay, like the ones that come from the back of our eyes, what we call the optic nerves, that carry the sensation of vision into our brain. There's also the opposite of those, what we call motor nerves, which are predominantly made up of motor neurons, and they carry signals out to cause contraction of, like, skeletal muscle, like the ones that control the way that our eyes move. What we call the oculomotor nerves, and allowing us to look up and down and around and all that wonderful stuff. I talked all about that in that previous lecture, in the video that I posted. As far as spinal nerves go, they're all like those mixed nerves. Like a two-lane highway, they carry information to and from the spinal cord. We designate these 31 pairs of spinal nerves based on where they arise in our spinal cord. We have our 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. In some regions, the axons from different levels of the spinal cord can come together to form what we call plexuses. It's basically like a tangled webbing of these spinal nerves that then give rise to nerves that can be quite large because they contain axons from multiple spinal cord levels. What I'm going to do real quick is show you an illustration from an anatomy lecture where I talk about this, which we haven't talked about in this class because we aren't to that point yet. But here is an illustration. Let me zoom just a little bit farther out. Oops, too far down. There we go. This is the illustration showing you the spinal cord. We can see that right down the middle. Okay, and as you see on the right side of the figure, there are bracketed out regions C1 through C8. You can see T1 through T12. Those are the names of the spinal nerves. Okay, we can see their pairs. There's one on each side. What's missing in this picture obviously would be the vertebrae, because we wouldn't be able to see spinal cord if the vertebrae were in there, because the vertebrae enclose the spinal cord. But I'm going to focus in right here and here to show off 2 plexuses. Cervical plexus is this kind of tangled network. Brachial plexus there in our kind of shoulder area is that tangled network. You can see where those nerves come off of the spinal cord, and then they kind of come together and create this network. That's what a plexus is. And then if you see under brachial plexus, we've listed off several nerves, okay, like the median nerve, radial nerve, ulnar nerve. Those nerves have been cut away, but they would run all the way down into the arm, like your ulnar nerve, which runs down over to like your pinky. Okay. Same thing for like the median and radial nerves that run down into your fingers as well. Okay. So really kind of more anatomy-based stuff, but I wanted to go ahead and talk about it because it is important to understand. As we know, there's big overlaps between these two classes because structure does affect function. Now, this illustration at the bottom was not the first time we've seen the structure of the spinal cord with the gray matter, white matter stuff and the associated what we call spinal nerves. As it states, and I've already mentioned this, each spinal nerve is a mixed nerve composed of both sensory and motor axons. In our illustration, we show, and I talked a little bit about this last time, just before attaching to the spinal cord, our spinal nerves branch to form what we call roots. One goes to the back of the spinal cord and is associated with the dorsal gray horn or the posterior gray horn. One that goes to the front of the spinal cord is called the ventral or anterior root and is associated with the anterior or ventral gray horn. Inside of the dorsal horn, we find predominantly sensory neurons, like this blue one here, which is going to be carrying a neuron impulse action potential into the back of the spinal cord, or a sensation like a touch or an itch or a tickle or a hot or cold, whatever. Okay. Remember, most sensory neurons we talked about are unipolar in structure, where the cell body's off to the side, right? You can see that right there in our picture. We talked about unipolar neurons in our first lecture of the unit. Okay. Remember, we don't, nerves are made up of thousands of neurons. So this illustration is kind of misleading because it looks like there'd just be one neuron here, a couple neurons in this root. There's going to be thousands. Okay. And remember, they're going to be held together in bundles. So all the axons will be together. The cell bodies will be together. And for that reason, actually, you can see that segment of the root is actually a little bit fatter than the rest. We call this the posterior or dorsal root ganglion. You can see it's labeled right there. That's because you've got a whole bunch of neurons with their cell bodies off to the side, pushing out. It's a little bit swollen right there. Okay. Well, they carry the signals into the back of the spinal cord where synapsing takes place with interneurons. Okay. We also see the other root, what we call the anterior or ventral root, which is made up predominantly, as it states, of motor neurons. Now, in this illustration, we show this red motor neuron being stimulated by an interneuron. And then the signal comes out, and as it states, being carried to action, or an action potential is being carried to skeletal muscles, which tells us right away that we're talking about a somatic motor neuron, because remember, somatic is always associated with skeletal muscle. Whereas in our green neuron, originating with the cell body in the lateral horns, we have an autonomic motor neuron that carries the signal out to our cardiac muscle smooth muscle glands, which we'll be talking more about here in a little bit. Okay. Let's see. What this illustration does not show, and we talked about this, and there was a little bit more sophisticated figure like this in our last lecture, is that not always does a signal come into the spinal cord and then just route right back out. When you're touched, for example, or when you're itchy, you have that conscious awareness of that sensation. We call that perception, which means that signal comes into the spinal cord, and then, and we talked about this last time, the white matter of our spinal cord acts like a elevator. It carries signals up and to carry signals down. Same thing for when we, wave our hand, or when you guys are taking your notes, we have to originate that signal somewhere, right? The cerebral cortex is the area in our brain that carries this, or is where that original signal starts. The primary motor area of our cerebral cortex, which we talked about last time, is where that starts using what we call upper motor neurons, which are found in the motor areas of our brain. We then carry the signal down, and then a synapse might occur or will occur with this somatic motor neuron or many somatic motor neurons that then carry the signal out to our skeletal muscles. So we can describe the functions of our spinal cord as kind of two major things, or there's two major functions. Okay, carrying signals up and down to and from the brain, but also, as it states here, for reflex activity. Okay, and we're going to talk about reflexes here in just a second. Before we do, let's talk a little bit more about anatomy associated with those spinal nerves. Okay, so let's talk about rami. A short distance after passing through its intervertebral foramen, a spinal nerve divides into several branches. We call these rami. Rami is plural, ramus is singular. So our illustration is looking down at a segment of the spinal cord, the spinal nerves, and then a vertebrae that is surrounding the spinal cord. You can see right in that hole within the vertebrae where you have the two branches that we just talked about, the roots, posterior root, anterior root, or dorsal and ventral, same thing, okay, where they connect to our spinal cord. Okay, right in this area, we see the spinal nerve as a singular nerve, but right after it passes out of our vertebral column, that spinal nerve branches again. So it branches right before it connects to our spinal cord, and then right when it leaves the column, it branches again. So when it's one kind of singular structure, it's like very, small and short, because it's branched on both ends. It's just kind of connected in the middle. Okay. The two roots, or two major roots I want to talk about, are the posterior and anterior ramus. Look at the size differential between the two. Okay, the posterior ramus is called the posterior ramus because it goes to the backside of our body, not the entirety of our body. Okay, it only innervates and forms nerves that include our muscles and skin of the posterior surface of our head, neck, and trunk. So think of like your back, what we would traditionally call our back of our neck, and our back of our head. That's not even, maybe a quarter of us, which, probably is pretty common sense why it's so small compared to the anterior ramus, which is going to supply the rest of our body. Okay, the posterior ramus has nothing to do with your legs, nothing to do with your arms. Okay, so your limbs, as it says, upper and lower, as well as everything in the front of our body. So that's a lot more, definitely more than probably a quarter of our body, considering we're not even including arms and legs here. So that's why this is more than 4 times the size of that one. Those those plexuses that I was showing you a little bit ago from that anatomy figure are going to be formed by the anterior rami predominantly, actually completely, not even posterior. Really, like I said, this is more anatomy based stuff, but it kind of is always important to kind of just have a brief review on it to get an idea of what we're looking at, what we're talking about. Now we can talk about reflexes. This is something we've mentioned a little bit about before. Talked about reflexes a little bit here and there the last week or so of class. And it is one of the most important things that our spinal cord does. And the other being what we talked about last time, acting like an elevator, carrying signals up and down from the brain. Our spinal cord can also promote homeostasis by serving as the integrating center for some reflexes, meaning this is where the decision making takes place, where we make a decision on what's happening to the body, and we decide what to do as a response. And we can describe reflexes as being quick, involuntary, stereotype reactions of glands or muscles to a particular stimulus. We know what quick means. It's super fast. We know what involuntary means. We can't control it. In fact, reflexes are, as it states, often taking place without our awareness and are difficult to suppress. We can't even it's hard to stop them. Okay, like putting your legs off the side of a table and having the doctor tap on that patellar tendon. You can try to hold your leg back, but what's it going to do? It's going to kick out, right? Okay, and that's really kind of what relates to the word stereotyped. I don't know, you guys are kind of young for, most of you guys anyway. This was before my time, but The Three Stooges was a TV show. It was popular way back in the day, where Larry, Curly, and Moe were known for doing stupid things. Okay, I don't know if it was an actual episode or if I'm making this up. I've seen it in a cartoon somewhere, if it wasn't that, where a guy goes to the doctor taps on the tendon, and instead of kicking the leg out, the doctor gets punched in the face. That doesn't happen in the real world. Okay? Doctors are testing reflexes because they know what's specifically going to happen. Okay? They occur in essentially the same way every time, and the response is very predictable. So why are they testing it? Well, sometimes they don't happen. And that tells a doctor when something's wrong. Okay? Now, we can classify reflexes a couple different ways. The first of which we see here on this page is based on where the integration takes place, whether it's in the spinal cord or if it's in the brain stem. If it's within the gray matter of the spinal cord, we call it a spinal reflex. That patellar tendon reflex that I'm going to talk about and have just mentioned is an example of one of those. Okay. Some of our more complex reflexes, related to like blood pressure and changes in breathing and that kind of stuff, are cranial reflexes, where the integration occurs within the brain stem rather than the spinal cord. Okay, but regardless if it's spinal or a cranial reflex, they share the same common characteristics. Okay, they don't just happen out of nowhere. They require stimulation, whether it's stretching of the muscle, tension on a tendon, stretch of the stomach or a blood vessel, something like that is going to be required. That's the sensory input. And then, as I've said, it's quick, generally involving few, if any, interneurons and minimum synaptic delay. Involuntary and stereotyped, I've already kind of talked about that. So those are two ways that we can classify reflexes. We can also classify reflexes based on the division of our peripheral nervous system that's involved. And that can be either autonomic or somatic. Autonomic, remember we said, think the term automatic, okay, because this involves the three effectors: cardiac muscle, smooth muscle, or glands. Okay. And so autonomic reflexes consist of contractions of those, and really should say, in my opinion, I just probably wrote word for word what was in your textbook, contraction, or it could also be relaxations of smooth muscle, cardiac muscle, or it could be secretion by a gland. We'll discuss these later. Okay. First thing, before I do that, let me touch on the term that's in there in parentheses behind autonomic, which is the term visceral. We haven't really seen that so much in this class, but in anatomy, we mentioned in our very first lecture that the term viscera or visceral refers to internal organs, whether that's your heart, your stomach, your liver, your spleen, bladder, or whatever. Viscera are those organs of our thoracic and abdominal pelvic areas. It could also be blood vessels. Really kind of aren't as thought of as as a organ, but are very important. For what we'll talk about here for the next several slides, we're going to talk mainly about somatic reflexes because they involve skeletal muscle, which is what somatic's always associated with, and they employ a reflex arc, which is where an action potential arcs into the spinal cord via a sensory neuron, or more than likely sensory neurons. We just show one here. We then decide what to do with that information, make a decision, And then the signal arcs back out of the same segment of the spinal cord, typically anyway, via motor neurons. There's 5 components to a reflex arc. It all begins with what's being stimulated, what we call the sensory receptors. For a lot of sensory receptors, it's just the dendrites of the sensory neuron. In this case, we are illustrating that with this neuron that has the dendrites. Looks like they're sensitive to pain when a nail or tack, whatever that is, penetrates the skin. So, we all know if you step on something like a tack, it's going to hurt, right? The receptors are the first component. They are going to respond to a specific stimulus, in this case, the pain of, a cut due to that sharp tack, which then creates an action potential. That's going to then travel by way of our sensory neuron. The second component carries that information from the receptors into the back of our spinal cord into what we call the posterior horn. Now, that's in the case of a spinal reflex. If this was a cranial reflex, the signal would be going up to the brainstem, where we would then have interneurons that decide what to do with that information. This is an illustration showing you a spinal reflex, so the spinal cord is involved. The interneuron acts as the integrating center, the point of synaptic contact between the 2 neurons in the gray matter of our spinal cord, or like I said, if it was in the brainstem, this is where we determine what to do. So the efferent or output neurons to issue a signal to the muscle. So this is going to decide what to do with that information. A signal travels out from the front of the spinal cord by way of a motor neuron. There's number 4 to our effector. In this case, since it's a somatic motor neuron, it's going to go to a skeletal muscle. If it was a autonomic reflex, it would not be skeletal muscle, it would be smooth muscle, cardiac muscle, or gland. And it'd be a little more complicated. Now, really, all this relates back to that very first lecture when we talked about feedback, right? There was 3 components to a feedback loop. You had sensors or receptors. Sensory receptor shown here. You had integrating center. There's number 3. And then you had effectors. So a reflex is an example of feedback. And we've been talking about this since the very first class period. Let's talk about different somatic spinal reflexes. The first of which we've actually already talked about. Now we just get to learn about it. All right, title of the slide tells you we're talking somatic, so skeletal muscles involved. Spinal tells you that the reflex is being integrated in the spinal cord. Does that mean the brain's not involved? No, it doesn't, because the brain will be involved. But the actual reflex itself, the spinal cord is in charge, okay? So I'll kind of make some sense of that here in just a second. First reflex I want to talk about is called the stretch reflex. It's called that because the initial stimulus is stretching of the muscle. Simple as that. Stretch reflex initiated by stretch of a muscle. Okay. Can be elicited by tapping a tendon that's attached to that muscle. And in response, what we're going to do is contract that same muscle. Tapping that tendon, like in this case, the patellar tendon, the knee jerk reflex, as we like to call it, is going to stretch your quad muscles. And it's not going to be very, drastic. Obviously, you're not, stretching them tremendously, but it will trigger this response. Okay. First thing we need to do is stimulate receptors. We have what are called muscle spindles in all of our muscles that wrap around the muscle cells or muscle fibers of our muscles. Their job is to monitor stretch. So when that muscle gets stretched, they become activated In other words, they depolarize the threshold, and action potential propagates through that sensory neuron into the spinal cord. Now, as you can see here, signal is going to go up to the brain. That's going to use the white matter. That's not involved with the reflex. What's it going to do? It's going to carry that signal up to the brain and give us conscious perception of that, feeling when the doctor hits you with the patellar tendon, or in the patellar tendon with the tendon hammer is what's called. We all feel it. That's touch. If they hit you hard enough, you'll feel pain. Okay. But that's not what we're talking about before the reflex. There's kind of two major things that happen in the spinal cord, as you can see here. Notice we've got some positive symbols. We've also got a negative symbol. These refer to excitatory for positive and inhibitory for negative. Okay. What we do is we synapse with a motor neuron, creating an excitatory signal in that motor neuron that travels back to the exact same muscle. If that excitation is strong enough, action potential rises in the motor neuron, propagates to the neuromuscular junction. That's what NMJ is, the point where the neuron meets the muscle, causing contraction of the muscle. That's our effector. And when you contract your quads, your leg will straighten out. You'll kick your leg out. Okay? We can only do that if we have the antagonistic hamstring muscles relaxed. Because your hamstrings are what bring your knee back into that flex position. If they are contracted at the same time, they balance each other out, meaning no motion is going to occur. So look what's happening here. We have an inhibitory signal. That's what these dotted lines are showing you, traveling to our antagonistic muscles, which causes relaxation in the opposing muscle. So these muscles are less likely to contract. This muscle contracts, and we respond with kicking of the leg. Now, in doing so, what happens to the stretch of the muscle? When a muscle contracts, it shortens. So there goes your stretch. So that's the whole purpose. We stretch the muscle so we shorten it. It's a simple, easy illustration of feedback, as we've talked about. Now, this is a commonly done reflex. Typically, it's done at the knee. You can also do it with tendons at the elbow, the wrist, and the ankle as well, though. And we call this type of reflex a monosynaptic reflex because mono means one. In other words, only one synapse is crossed in our central nervous system. Essentially, the exact opposite of that reflex is the tendon reflex. The tendon reflex is a feedback mechanism that controls muscle tension. So the last one was stretch. This one controls tension by causing, instead of a muscle contraction, it causes muscle relaxation before the muscle force becomes so great that the tendons might be torn. Okay, so in this case, what we're doing is we're contracting the quads super, super strong, super, super hard. Okay, we're like trying to kick our leg out against the concrete wall, something like that. We can't push the wall over. We're contracting really hard, and tension's going way up in the muscle, which puts a tendon at risk for being damaged. Same thing for the muscle. We have receptors in our tendons called Golgi tendon organs, or just tendon organs, or GTOs. I've seen them listed all sorts of different ways. But their job is to detect changes in muscle tension caused by passive stretch, or in the case of what I've been talking about, muscle contraction. So tension is really high. They detect that. An action potential is created that then propagates into the back of the spinal cord, just like we saw last time. Signal goes up to the brain for perception, not so important for the reflex. What is important is taking place in the spinal cord. This time we send an inhibitory signal to that same muscle that the tendon's attached to. The muscle that's doing the contracting is inhibited. And if you inhibit a muscle, it reduces its contraction. And that's going to relieve that tension. Also, to help relieve that tension, we excite the antagonistic muscles. The hamstrings now contract, pulling the leg back, which causes the muscle to relax even more. Thus relieving the tension on the tendons. This is a, like I said, preventative reflex to prevent tension and force becoming so great that tendons might be torn. Now you might be wondering, well, what about like when you lift weights? Okay. I said a little bit ago that it's difficult to suppress reflexes. You can kind of suppress this one. This is what you have to do if you want to lift weights. Because anytime you would be lifting weights, especially if it's really heavy weight, you're going to activate this reflex, which means that the muscle would relax and you wouldn't be able to lift anything. And so lifting something heavy, you can kind of actually suppress this. That's kind of a cool thing we can do. Our next two reflexes I want to talk about kind of simultaneously because these actually happen simultaneously. And these are the flexor withdrawal reflex and the cross extensor reflex, both of which take place whenever we have a painful situation arrive. Well, let me rephrase that. Whenever we have an acute painful situation, like as it shows on the next page, stepping on a tack. Now this is illustrating just the flexor withdrawal reflex. The flexor withdrawal reflex, it's kind of what I've been talking about. I use the analogy or the example of touching the hot stove. Same thing's going to happen in that situation. You step on something sharp or you touch something hot, what do? You remove that limb or hand or foot from that painful stimulus, right? Okay. That means you have to withdraw, in this case, the leg by flexing. That's why it's called a flexor withdrawal reflex. Okay. So once again, begins with stimulating receptors, in this case, the dendrites of a sensory neuron in our toes. Action potentials propagated up into our spinal cord. Now in this illustration, we show not one, but three segments of the spinal cord because this is an example of an intersegmental reflex arc because it involves multiple segments of a spinal cord. So we actually come into the spinal cord and we also send signals up and down the spinal cord. The same response is going to come from all three levels. In this case, an excitatory signal to, in this case, the hamstring muscles. Each of those signals activates a different segment of the hamstrings. Thus, the entire muscle is essentially contracting. So it's a quick, strong reflex, withdrawing the leg, pulling the leg really quickly away from that painful stimulus. Now, while that's taking place in the right leg, we also have what's called a cross extensor reflex taking place in the opposite limb, which is what's shown over here on the left side, or excuse me, the right side of the illustration on this picture. Okay. From this side, we can see this is the flexor withdrawal reflex. What we just talked about, signal comes into the spinal cord, we go up and down, signal goes back to cause contraction of the right hamstrings. If you step on something sharp, something hot, whatever, you withdraw your right leg, what do you have to do with your other leg? Straighten it out so you can shift your body weight and lift your right foot off the ground, right? Because if you didn't have a left leg to switch your body weight to, what's going to happen? It's going to fall over. Okay? So on the opposite side of the body, where that signal came in to the spinal cord, we cross over from one side of the spinal cord to the other. Signals go up and down. Ultimately, the same response occurs from each of those levels. A signal comes out the front of the spinal cord. That goes, in this case, to the left quad muscles, which is going to extend that limb, making it a straight leg for us to shift our body weight to. Pretty cool. Look at all the neurons that are involved in something that we experience, you know, maybe not every day, but I do, because I've got Legos and army guys and all sorts of stuff all over my floors. And I'm stepping on stuff all the time. The next time you step on something, just think about what all is taking place. Anytime you see neurons meeting, remember all those steps to a synapse where the voltage-gated calcium channels open, synaptic vesicles release the neurotransmitters that cross the cleft. Remember the voltage-gated sodium channels and the potassium channels that open and close every time a action potential is propagated. So I say that because I hope you, A, understand how damn complicated the nervous system is, but also B, just grow to appreciate how amazing it is because of its complexity and how amazing, quick, and just insanely intricate all of this really is. Should I say that? Obviously you know that because I imagine you've been studying all that stuff. Hopefully anyway. I've already realized. Okay, we're going to be moving on from reflexes to really, at this point, focusing on the autonomic nervous system, whereas we were just kind of focused on the somatic, which once again is one of the two major divisions of our peripheral nervous system. Somatic, we know, is associated with skeletal muscle. Essentially, we can describe it as the division that controls our skeletal muscle, specifically in a, for the most part, voluntary manner, done with our conscious, purposeful intent. Whereas, as we know, the autonomic, I always say automatic, associated with controlling our glands, our skeletal, or excuse me, our cardiac and our smooth muscle, and it's done involuntarily without our conscious intent or awareness, as it states there in the middle of that paragraph. The autonomic nervous system is composed, as we know, of sympathetic and parasympathetic motor neurons and brain regions that regulate their activities. We're going to talk a lot about sympathetic, parasympathetic here for the rest of the lecture, for the most part. Okay. These two divisions, sympathetic, parasympathetic, have the ability to innervate the same structures. Okay, for example, we know the sympathetic, which produces a, what is that response called? Fight or flight, is going to do what to your heart rate? Make it go up. Whereas the parasympathetic, which produces a rest and digest, is going to make your heart rate go down. They innervate the same organs. We call this dual innervation. And that means they innervate the same organs, but they have contrasting effects on those same organs. Now, we don't change our heart rate just willy-nilly out of nowhere. We don't change our breathing rate willy-nilly out of nowhere, and so on and so on and so on for any and everything that sympathetic, parasympathetic do. Just like we don't kick our leg out for no reason and have these reflexes happening that we just talked about for no reason. We all have to, or all these reflexes, all these responses require a stimulus. Okay, for input, which is what stimuli create, Input to the ANS comes from autonomic sensory neurons, which are primarily associated with what we call interoceptors. We call them interoceptors, think of because they're located in our interior, in places like our blood vessels, monitoring the stretch, or the blood pH and blood oxygen levels, monitoring the stretch of our stomach, pH of our stomach, calcium levels, and stuff like that. We've got them all sorts of different places. Besides our blood vessels, our viscera. We've got them in our muscles. We even have receptors in our brain and other parts of our nervous system monitoring their conditions as well. And those are all things, well, obviously, in our internal environment. We'll talk a lot more about interoceptors, actually not a lot, but more about interoceptors when we talk about sensations. That's the very next lecture. We'll get to that in unit number 3, which is our next unit, when we are focusing on all the different sensations of the body.
How
This
does this affect like people with tics and stuff?
like a motor tic, like, you know, when you're twitching and stuff like that, or something. I don't know how all that works.
I was wondering, like, because like certain medications can cause you to have,
Like the jitteries.
yeah, or
I don't know exactly what all goes into that. That's a good question. Maybe you can do some independent research for us. All right, let's talk about those outputs of our autonomic nervous system. Autonomic motor neurons regulate their visceral activities, or visceral activities, not there, visceral activities by either increasing, which we could say exciting instead, or decreasing, which we could say inhibiting, ongoing activities in their effector tissues. And once again, the effectors associated with the autonomic nervous system: cardiac muscle, smooth muscle, and glands. So we're either going to activate or inhibit, essentially those different things. Okay, excite, inhibit, increase, or decrease, whatever you want to say. Okay. As opposed to somatic motor neurons, which are usually myelinated and travel all the way from the CNS to the skeletal muscle fibers that they innervate. Autonomic motor pathways consist of 2 motor neurons in series. Let me go back to this very first page of our lecture. I didn't talk about it at the time, but illustrated here at the bottom is showing you the somatic nervous system innervating skeletal muscles, or a skeletal muscle in this case. When we contract our skeletal muscles, the signal that activates those specific muscles actually originates up in our brain with the cerebral cortex. The very last leg of that signal travels all the way from our brain or spinal cord, in this case spinal cord, from the anterior horn where that cell body is, or the cell bodies of those neurons are, all the way to the muscle. So when you curl your toes and you're contracting muscles all the way down in your leg and foot, you're actually carrying a signal from your spinal cord, from your lower body, your lower back, all the way through your legs and into your feet. Which means those neurons are super long, right? And whereas when we carry a signal to your heart, to maybe a salivary gland, I think that's what they're showing here, or like the bladder, which is what this is, we actually carry a signal from not just 1, but 2 neurons before we get to those effectors. This is showing you the autonomic nervous system. So here with somatic, there's 1 neuron. Here we have 2. Those two neurons are called preganglionic neurons and postganglionic neurons. If you break down the word preganglionic, postganglionic, pre we know means before, and post means after. Okay, what ganglion are is, and I always say this, and I really should put it in air or quotes here, points of contact, because as you know, they don't actually touch. There's a little gap in there where neurotransmitters are released, like the ACH, acetylcholine, that we see in our two figures. The preganglionic neuron is the neuron that comes first before that point of contact, and then the postganglionic neuron comes after. The first neuron is the preganglionic neuron, where the cell body is located in the central nervous system. Either the spinal cord, as it shows here, or it could also be from the brain. And then the postganglionic neuron is the one that actually directly innervates the cardiac muscle, smooth muscle, or glands. As we know, there are two divisions to the autonomic nervous system. For the outer output or motor part anyway, the parasympathetic, sympathetic, which I've already mentioned a little bit about here this afternoon. Let's continue talking about sympathetic, parasympathetic. As we've already stated, sympathetic nervous system produces that fight or flight response, so we can describe it as a fight or flight division. And that means that it adapts our body for physical activity. To either fight or flight, run away. Can be drastic, like a bear chasing us. Could be very subtle, like just, getting up and exercising. Running around the building, doing jumping jacks, whatever. Okay. Doing everything in its power to make us capable of doing physical activity, increasing heart rate, blood pressure, increase our pulmonary airflow, blood glucose levels, and blood flow to cardiac and skeletal muscle. We'll talk a lot more about specific responses. On this next page, we show a comparison between sympathetic on the right and parasympathetic on the left. I'll talk about the sympathetic first. All output for the sympathetic nervous system comes from this segment of our spinal cord, from T1 to L2. If I go back to this illustration here, talked about this figure the other day, and we said that in only certain segments of our spinal cord do we find lateral gray horns. Said it was between T1 and L2. That's where you find the cell bodies of the, and it could say autonomic motor neurons. In parentheses, it really should say sympathetic division. If we're talking about T1 through L2. Because that's where the output comes from. That's where the cell body of those first, what we call the preganglionic neurons, are located. Now, this illustration is only showing you one, but there are two of these, we call them chains or trunks. They're on the outside of our spinal column. Spinal column's not shown, so we can show the spinal cord. We'd have another one here. But typically what we see is the signal leaves the spinal cord, and then we see a second dot right there in that chain, like we see them up here as well, where the first neuron synapses with the second neuron, which is the postganglionic neuron. So typically, the first of the two neurons is very short. Okay. The preganglionic neuron is very short. The second, the postganglionic, is very long. which is illustrated here in this figure at the bottom of that previous page. A short first and a long second. Since the thoracic and lumbar spinal cord are involved with the sympathetic nervous system, another term to describe the sympathetic division is the thoracolumbar division. Thoraco for thoracic, lumbar for obviously lumbar. Looking at this side of our figure now, we don't show any activity for that segment of our spinal cord with regard to the parasympathetic nervous system. Instead, output for the parasympathetic actually comes from two different distinct parts. Cranial nerves and sacral spinal nerves. Okay. Specifically, 4 cranial nerves shown here. We talked about those once again on, well, we didn't actually, but I recorded a video last Thursday, the very end of our last lecture. And then the sacral region of our spinal cord. Okay. This time, the first neuron is really long. The postganglionic, the second one, is really short. So it's a reverse of what we see for the sympathetic. This one is short and then long. This one's long and short. We also don't utilize at all the chains. The trains or trunks that we talked about with the sympathetic are not involved with the parasympathetic. Since we're dealing with cranial nerves in the sacral regions of the spinal cord, we have another term to describe the parasympathetic, just like we did with the sympathetic. We call it the craniosacral division. So you want to know that the sympathetic is also known as thoracolumbar, parasympathetic is also known as craniosacral. Now, when I first introduced sympathetic, parasympathetic, I talked to you guys and explained how they work kind of like a teeter-totter, where they have balance between the two, meaning if one goes up, the other one's going down. And I kind of demonstrated this and how it can turn on a pretty quick dime, where I said, you know, right now you're illustrating a rest and digest response. You're not breathing rapidly. Your heart rate's pretty low. Blood pressure hopefully is low, so on and so forth. Then I said, what would you do if I said, let's take the exam right now? And, you know, people got a little worked up, even just me saying that, because, you know, their fight or flight response was going to be kicking in, where heart rate's going to go up and blood pressure is going to go up and all those wonderful things that we've all experienced whenever we had that kind of shot of adrenaline taking place, right? This balance between the two is called autonomic tone. Something I want to know. Now, I've been talking a lot and illustrating different things with figures. I really like the 4 figures that are on this page to show you. These are essentially what I've already been talking about. Somatic at the top, which you'll probably want to highlight the fact that that's somatic, showing you that we've got one long neuron that travels all the way to the effector being skeletal muscle. I would, in your lecture, bracket off these 2 middle pictures and label them as the sympathetic nervous system. And then I would draw an arrow and point to this figure, the 4th one, and label that as the parasympathetic nervous system. I just said something, and I did it purposefully, that we've all experienced a shot of adrenaline, right? We know what that experience is like. That takes place whenever the sympathetic nervous system is activated. And I was just talking about how whenever the sympathetic nervous system is activated, it arises from the thoracic and lumbar regions of our spinal cord, comes out of the spinal cord via a preganglionic neuron. We then synapse with a postganglionic neuron that goes the rest of the way to the effectors. What the illustration on this page was not showing you is that something else is also taking place whenever the sympathetic nervous system is active. We're innervating the adrenal medulla, which is the core of our adrenal glands, where we store and release epinephrine, norepinephrine, and a very small amount of dopamine. Predominantly, what we're going to focus on is epinephrine, which we know best as adrenaline. And we dump that adrenaline and norepinephrine into our blood as hormones, which can then be transported to many places in the body, wherever blood goes in our body. Yep. Whereas whenever we're using the parasympathetic, we use that long preganglionic, short postganglionic, and we're not involving the adrenal medulla. Now, I'm already getting ahead of myself because I talk about all that stuff on this page. So go ahead and write it out, and then I'll read through and talk about it. Oh, well, there we go. Okay, reading through. Most preganglionic neurons of our sympathetic division synapse with postganglionic neurons in this sympathetic chain of ganglia. Here, this illustration is essentially showing you what we showed on this page, just this part, but now we show two sides. So we're showing two chains because there are two chains. Okay, not the wrapper two chains, but the two chains or trunks. Okay. The synapse typically takes place within the chain. Not always, because sometimes the signal travels through because the preganglionic neuron then goes out to what we call collateral ganglia and then synapses. Okay. Closer to the digestive tract, but overall the sequence of events is still the same. First ganglionic or preganglionic, first neuron, second neuron, postganglionic, then we have the effectors. What's cool about the chains is they can allow signals to go up and down because some neurons, as we know, have right turns to them, what we call axon collaterals. Okay. And that's what I'm talking about right here in this bullet. Preganglionic axon collaterals have the ability to travel up and down the chain to synapse at different levels of the chain of ganglia, because we have to synapse, or we have to send signals to our face to innervate things like our salivary glands and our eyes and things in our brain. Well, if we're coming out from the thoracic and lumbar region of the spinal cord, we have to travel up somehow. These are like little elevators for the signal to go up and down. Okay. One exception to this basic sequence, what I was just talking about on the previous page, involves the adrenal medulla, an inner region of our adrenal glands, where preganglionic neurons synapse in the adrenal medulla just as if they were part of the sympathetic chain of ganglia. So as you can see, the first neuron still comes. There's just not a second neuron. Instead, we have what we call chromaffin cells that are activated and release epinephrine, which is, as we know, adrenaline, norepinephrine, and then a very small amount of dopamine. Not even a measurable amount. Predominantly, though, as I show you there, 80% of the secretions from the adrenal medulla will be epinephrine, 20% norepinephrine. And they are released as hormones because they're dumped into the blood, whereas, you know, in other places where we'll see norepinephrine, it's a neurotransmitter because it's released by a neuron. Okay. These hormones will then circulate in our body, intensifying responses elicited by our sympathetic nervous system. We'll talk about those responses here after a bit. Unlike the sympathetic nervous system, our parasympathetic nervous system, PSNS, does not have ganglia. It really, I guess I did fix it. I used to say that are not in a chain. It does say it perfectly now. I took out the second not. Does not have ganglia that are in a chain, so you don't see the chains involved here. Instead, we have what are called terminal ganglia, which are shown here, kind of blue ovals, where the synapsing takes place. As far as output for your parasympathetic nervous system, the majority, about 80% of our parasympathetic activity comes from our paravagus nerves, cranial nerves number 10. And I talked about these, and I talked about this in our last couple slides of our last lecture that I recorded and posted over the weekend. If you haven't watched them, make sure you do. The term vagus, and I talk about this in the video, is not spelled like Las Vegas. The AGUS translates to mean wandering. Remember, these are cranial nerves, so they are attached to your brain. Look where they go. All the way down to your stomach and intestines, liver, lungs, heart, all are innervated by those pairs of nerves. It means that they travel down through your neck, down through your chest. They go through the diaphragm into your abdomen. They're wandering nerves. They're really long, very highly branched nerves. The cell bodies of these preganglionic neurons of our vagus nerves are located in our medulla. Their axons extend into the organs of our thoracic and abdominal cavities. For the other ones that you see, cranial nerve 3 and 7 and 9, they stay more so close to the brain, like in your face, innervating things like your salivary glands and lacrimal glands and stuff like that. Whereas your most inferior organs, like those of our reproductive system, our bladder, and other structures of the urinary system, they're going to be innervated by the sacral region of the spinal cord, which should make sense because they're close by our sacral spinal cord. And so there's where the sacral outflow goes, as you can see in our bottom of the picture. So as noted previously, our sympathetic and parasympathetic nervous system have what we call dual innervation, meaning they innervate many of the same target organs. Using the heart as an example, we know our heart is innervated by both sympathetic and parasympathetic neurons. Well, how the hell does our heart know to speed up when we have a fight or flight response, or to slow down when it's a rest and digest response? It all relates back to the neurotransmitters that those two divisions secrete. They choose to release different neurotransmitters, and organs have receptors for those different neurotransmitters. Now, we've talked a little bit about neurotransmitters, tail end of our nervous tissue lecture. We mentioned acetylcholine, and we're going to talk acetylcholine again here in this lecture. Acetylcholine, I've had a professor pronounced it acetylcholine, however you want to pronounce it, is abbreviated capital A, capital C, lowercase h, released by cholinergic fibers or cholinergic neurons, cholinergic because acetylcholine. And those neurons include all sympathetic and parasympathetic preganglionic neurons. No matter what, if you are looking sympathetic or parasympathetic, which we showed here in this figure, or if we go back to this figure, the first neuron, even if it's sympathetic or parasympathetic, is always releasing, as it shows, acetylcholine. The second neuron for the parasympathetic is also going to release acetylcholine. So when you are dealing with the parasympathetic nervous system, it's acetylcholine and then acetylcholine. Here's where the difference is. Look at the sympathetic nervous system and what it releases with its second neuron. Not acetylcholine, it's NE, norepinephrine, unless we're talking about postganglionic neurons of the sympathetic nervous system innervating our sweat glands. For whatever reason, sweat glands are always going to be activated by acetylcholine from the sympathetic nervous system. And that's what this illustration is showing you. So this is sympathetic preganglionic releasing acetylcholine, sympathetic postganglionic also releasing acetylcholine. For anything else, we'll talk about this here in a second. The second neuron of the sympathetic nervous system is going to release norepinephrine. Now, if I go back to this illustration again, acetylcholine, as you can see, is also released by the somatic motor neurons that innervate our skeletal muscle. So if on the exam you're wanting to know and you don't know the answer, I ask you, this neuron is secreting what neurotransmitter? Odds are in your favor. Hopefully you do know, but when in doubt, acetylcholine is a pretty dang good answer. And that's why I said it's the most common neurotransmitter in the body. We release it a lot of places. Okay. All sympathetic, parasympathetic, preganglionic neurons, all postganglionic neurons are parasympathetic, and then sympathetic postganglionic neurons innervating our sweat glands. Receptors for acetylcholine are called cholinergic receptors, and there's two types: muscarinic, found on the effectors, cardiac muscle, smooth muscle, and glands, and then nicotinic, which are found on the dendrites and cell bodies of the sympathetic and parasympathetic postganglionic neurons, the chromaffin cells, the adrenal glands, and at the neuromuscular junction. So muscarinic, activated by acetylcholine. Muscarinic up here, even though it's kind of cut off, you would be able to see it's muscarinic here. Nicotinic are found on the dendrites and cell bodies of neurons in all three pictures. We also, as I've already been talking, involve another neurotransmitter, norepinephrine, secreted by nearly all sympathetic postganglionic neurons, the exception being the ones that innervate our sweat glands. Neurons that secrete norepinephrine are called adrenergic fibers or adrenergic neurons. And I just mentioned a little bit ago about norepinephrine, and that it is released by our adrenal medulla as a hormone. If it's dumped into the bloodstream from our adrenal medulla, it's a hormone. If it's released by a postganglionic neuron, it's a neurotransmitter. Is there any difference in the chemical? No. If it's released by a neuron, it's a neurotransmitter. Released by a gland, it's a hormone. Simple as that. Same chemical, it's just where it's going, it's different. Okay. And that's what I'm talking about right here. Can either be released as a neurotransmitter by sympathetic postganglionic neurons or as a hormone from the adrenal medulla. Epinephrine, whereas is released as a hormone into the blood. Receptors for norepinephrine and epinephrine are adrenergic receptors. We find them on visceral effectors innervated by most sympathetic postganglionic neurons. And there are two types. It might look like A and B, but that's actually an alpha sign and a beta sign. Alpha, adrenergic. Alpha kind of has an extra little tail at the top and the bottom, looks like an A. Usually those produce excitatory effects like causing blood vessels to constrict. When I say excitatory, when it comes to muscles, when there's an excitatory signal in a muscle, it contracts. Whereas in an inhibitory signal, it's going to cause the muscle to relax. And in betas, which you can see looks like a B, but beta has an extra little tail at the front, they can have differing effects in different parts of the body. For example, we want to be able to breathe in more oxygen and out more CO2 in our lungs, which means we want the airways to be more open. And we can do so by relaxing the muscle in our airways, which is an inhibitory signal. Whereas when we activate a stronger and harder or stronger and faster heartbeat, that's an excitatory signal involving beta 1 receptors. Does that make sense? So it's all just dependent upon where and what the response that we want to have happen. Now, as far as these pictures go, I actually am really wanting you to pay attention to them. Especially not so much this set, but these 4 images. You should be able to look at those 4 images individually. Tell me which division of the nervous system is active. So somatic, sympathetic, parasympathetic, and then be able to identify which neurotransmitters are released. Like I said, when in doubt, acetylcholine is a pretty good answer. It's not a foolproof thing because, as you know, norepinephrine, epi and norepi here. Everywhere else, though, it's acetylcholine. I might put a picture or 2 on the exam. Let's talk about neurotransmitters and drugs. A large variety of drugs and natural products can selectively activate or block specific cholinergic or adrenergic receptors. So in other words, we can essentially have drugs, now these can be illicit drugs or medicine type drugs, that can basically act like a neurotransmitter and bind to a receptor, producing a response, or they can block the receptor and thus prevent the neurotransmitter from binding to that receptor and prevent a response. Okay. An agonist is a substance that binds to and activates a receptor. Then, in other words, it mimics the effect of a natural neurotransmitter or hormone. The example is phenylephrine, a nasal decongestant, which is going to be found in just about any cold and allergy medicine that says it's a nasal decongestant. What it does, it constricts blood vessels in our nasal mucosa, the inner lining of our nose, reducing mucus production and thus relieving nasal congestion. So next time you take some medicine to clear your nose, and what I'm talking about, look for phenylephrine on that. I imagine you'll find it. You guys ever heard of Mucinex before? It's got it. An antagonist is a substance that binds to and blocks a receptor, which prevents the natural neurotransmitter hormone from exerting its effect. Example would be a class of drugs called beta blockers, often prescribed for people with hypertension, high blood pressure. This is fancy, or is what hypertension means. They block the beta receptors, hence the name beta blockers. And they bind to all beta receptors, prevent their activation by epinephrine and norepinephrine. The desired effect is the blockade of specifically the beta 1 receptors, thus decreasing heart rate and force of contraction, which thus results in a decrease in blood pressure. Anybody heard of beta blockers before? Anybody that's got high blood pressure has had any heart problems, there's a good chance they're going to be put on beta blockers. Okay, not quite done talking sympathetic parasympathetic, but we're getting close. Here is a table, or not a table, but a set of things kind of listing off different responses that our sympathetic nervous system produces. Once again, we don't have a fight or flight response activated without a input, right? A stimulus, some type of stress. And we'll get into stress more in our next unit when we talk about the stress response and hormones that are involved. But stress increases activity of the sympathetic nervous system, activating that fight or flight response. Once again, we need energy. So we increase production of ATP, dilate our pupils so we see better, increase our heart rate and blood pressure so that we can circulate more blood, dilate our airways, breathe in more oxygen, breathe off more CO2. We constrict blood vessels supplying our kidneys and GI tract because it's not important to digest food to make urine if we are in a fight or flight response. And we can then route that important blood to the things that are working, things like our skeletal muscles, our cardiac muscle, our liver, and our adipose tissues. Because in our liver, we have glycogen, the stored form of glucose that we can then break down, a process that's called glycogenolysis. We can then use that glucose that's released into the blood to make ATP, just like we can do with fat through the process of lipolysis, the breakdown of fat for ATP production. Whereas we have essentially a lot of the opposite things taking place with the parasympathetic nervous system. When we have that, as we say, rest and digest response after. If you can remember SLUD, it's not like a word, but SLUDD, those are kind of the 5 principal things that we do during a rest and digest response. We all hopefully know what salivation is, production and release of saliva. Probably the only one that you don't know, maybe so much, is lacrimation. Anybody know what that is? Your lacrimal glands produce and secrete your tears. Well, we're not crying all the time, but we are keeping our eyes moist, right? We're always producing a little bit of lacrimal fluid to keep our eyes moist. If we don't, we have to get some eye drops as our eyes dry out. Urination, we all know that's a fancy word for peeing. We all know defecation is a fancy word for pooping. Digestion is the breaking down that leads us to be able to poop. In other words, we're conserving or storing body energy. Digestive and urinary functions are elevated. Body functions that support physical activity are decreased. Those are very brief, easy descriptions of sympathetic and parasympathetic responses. Table 15.4, you can see, continues on to a couple slides, goes through really specifically what many, probably all the organs, I've never actually read through all of them, what it's going to do for them. Sympathetic is the middle column, parasympathetic is the far column on the right. These tables I'm not going to specifically ask you to know. It's a lot of information. Here's something I would want you to know, though. It's a pretty simplified version of that set of tables. Be able to look at this figure, shows on the left what the parasympathetic does to things like your eyes and your heart and your genitalia. Sympathetic on the right. Same thing for all those various organs. A lot of them hopefully are common sense. If you can remember, sympathetic is the fight or flight. Parasympathetic is the rest and digest. It'll probably go a long ways. There will be several questions on your exam. Obviously, we've spent a lot of time today talking fight or flight, rest and digest. Unfortunately, I'm just giving you a heads up now, this isn't going to be the end of talking sympathetic, parasympathetic. We're going to talk about it all semester long because we're going to talk about blood pressure and breathing and digestion and all those different systems of the body. And remember, the nervous system is involved with all of them because the nervous and the endocrine systems are in charge for the rest of it. So learn it now. It'll be helpful later on. Last thing I want to talk about are autonomic reflexes in the autonomic nervous system a little bit more. Autonomic reflexes are responses occurring when nerve impulses pass through an autonomic reflex arc. And these play key roles in regulating very, very important things. Things like blood pressure and digestion, urination, stuff that we'll talk about a lot in the next month and a half or so that we have class. Same 5 components exist to a autonomic reflex arc as I did with a somatic reflex arc. We still have sensory receptors. Typically, these are interoceptors because they're in our interior, whereas a lot of our somatic reflex sensory receptors were in our skin or muscles or tendons. So we saw for like stretch and tendon reflexes. We still have a sensory neuron that then propagates the signal into our central nervous system. In other words, to our integrating center. Now, for the main integrating centers of our autonomic nervous system, we're talking mainly about the hypothalamus and the brainstem. In our last lecture, we talked about, just how important our hypothalamus was. We talked about the medulla, that's part of our brainstem, pons, that's part of our brainstem, so on and so forth. Some autonomic reflexes, though, like urination, defecation, for example, the integrating centers are in the spinal cord. Notice here, for motor neuron, I actually pluralized it because, remember, for motor neurons of the sympathetic, parasympathetic, there are 2, a pre and a post. Now, we aren't specifying at this point whether this is sympathetic or parasympathetic output, but the same thing would be seen regardless if it was sympathetic, parasympathetic, a pre and then a post. That then travel to the effectors, which once again, effectors of our autonomic nervous system are always going to be 3 things: smooth muscle, cardiac muscle, and glands, which elicit a change. Typically, autonomic reflexes and changes elicited by our autonomic nervous system are done without our conscious perception. We are not even typically aware when our blood pressure changes or our oxygen levels go up and down, and we fix them and stuff like that. As I just mentioned, the major control and integrating center of our autonomic nervous system is the hypothalamus. Once again, we cannot have reflexes without input first. So input for visceral functions, olfaction, gestation, as well as input for changes in temperature and blood osmolarity, which is a fancy way to describe water levels and levels of various substances in our blood. But also, remember the limbic system? Talked about that last time. Remember the nickname for the limbic system was our emotional brain. We talked about how emotions can result in changes to our internal organs. How, for example, we might, just think about somebody that we don't like and how that can cause us to become agitated, and our heart rate's going to go up, and we'll sweat, so on and so forth. Those are things where the input's a little bit trickier. It's coming from the brain itself. But then the hypothalamus results with a output information to influence autonomic nervous center, or autonomic centers in our brainstem and spinal cord. What's kind of cool is our posterior and lateral aspects of the hypothalamus control the sympathetic division, whereas the anterior medial parts control the parasympathetic. Any questions?